Hello and welcome back to OncoTrial Insights with Dr. Pidugu, where we bring the latest updates in oncology clinical trials for everyone. I'm Megan, and joining me is Dr. Pidugu. Today, we're diving into some exciting and promising results from the JAMA Oncology paper, Ensartinib versus Crizotinib for patients with anaplastic lymphoma kinase-positive non-small cell lung cancer, a randomized clinical trial. This pivotal study led to the FDA approval of Ensartinib for the treatment of ALK-positive locally advanced or metastatic non-small cell lung cancer. Welcome, Dr. Pidugu.
Thank you for having me, Megan. And thank you to our listeners for tuning in. Before we get started, I'd like to share a brief disclaimer. The views and opinions expressed on this channel are solely my own and do not represent those of any institution, organization, or employer. This content is for educational and informational purposes only and should not be considered medical advice. Always consult a qualified healthcare professional for medical guidance. The medical information on this site is provided as information only and is not to be used or relied on for any diagnostic or treatment purpose. Please consult your healthcare provider before making any healthcare decision. The contents and other forms of communication such as text, graphics, images, and other material created by this channel are for information purposes only. The information I will discuss is not meant to endorse or criticize any specific products or brands. My goal is to provide you with educational content, shedding light on various topics. What you're about to see is purely for educational purpose only. Now, with that said, let's dive into Ensartinib versus Crizotinib for patients with anaplastic lymphoma kinase-positive non-small cell lung cancer, a randomized clinical trial, and what this trial means for cancer care.
So let's zoom in on what's really happening at the molecular level with ALK-positive lung cancer.
Sure, let's talk about a major driver in non-small cell lung cancer, NSCLC, ALK gene fusions. NSCLC is the leading cause of cancer-related deaths worldwide. ALK stands for anaplastic lymphoma kinase, a receptor tyrosine kinase involved in the development of the nervous system. In healthy cells, it's tightly regulated and not normally active in adult lung tissue. But in certain non-small cell lung cancers, a chromosomal rearrangement fuses the ALK gene with their partner gene, most commonly echinoderm microtubule-associated protein like 4, EML4. This creates a chimeric fusion protein like EML4-ALK. Here's the problem. This fusion protein is constitutively active, meaning it's always on. It dimerizes without ligand binding, triggering continuous activation of ALK tyrosine kinase domain. This leads to persistent downstream signaling through pathways like Ras-Raf-MEK-ERK drives cell proliferation, PI3-KA-KT-mTOR promotes survival and growth, and JAK-STAT supports immune evasion and growth. The result? Unchecked cellular growth, survival, and metastasis, a perfect storm for cancer.
Let's talk about how ALK inhibitors work.
Absolutely. Targeted therapy aims to interrupt this abnormal signaling. First-generation inhibitor, Crizotinib, blocks the ATP binding pocket of the ALK kinase domain. Initially effective but incomplete inhibition and off-target effects mean many patients relapse within 1 to 2 years. Second-generation inhibitors, Ensartinib, Alectinib, and Brigatinib, more potent and selective, can inhibit many of the ALK point mutations that arise under Crizotinib pressure, such as L1196M, C1156Y, and G1269A. But over time, more resistant mutations develop, especially G1100R, which creates steric hindrance in the ATP binding site. Third-generation inhibitor, Lorlatinib, designed to overcome the toughest resistance mutations, including G1202R and even compound mutations. The bottom line is that ALK fusion protein hijacks cellular signaling, but we've developed a smart lineup of inhibitors that progressively tackle evolving resistance mechanisms. It's a real-time arms race between cancer evolution and drug development.
Okay, so walk us through the clinical trial EXALT-3 study. What was the setup?
Sure thing. So it was a randomized open label phase 3 trial. Now, what that means in simpler terms is patients were randomly assigned to get either Ensartinib or Crizotinib, and everyone knew which treatment was being given. No hiding it.
Transparency is key.
Absolutely. This is an open label, randomized, multicenter, active control trial. The way it was set up with a good number of patients, 290 patients, all with advanced ALK-positive NSCLC, and importantly, none of them had gotten any ALK inhibitor treatment before. 120 of them spread over 21 countries between July 2016 and November 2018, and all this happening across a whole bunch of centers. They were randomly assigned to receive either Ensartinib or Crizotinib. The primary goal was to see how long patients lived without their cancer progressing. That's what we call a progression-free survival.
So, fresh slate and global effort, huh?
It really was. It paints a pretty solid picture of how these treatments compare in the real world.
A solid picture. I like that. Okay, so the big question, what were the researchers laser focused on? What was the gold standard, the measure of success here?
The primary endpoint, the main outcome they were monitoring, was how long patients lived without their cancer getting worse. This is known as progression-free survival. To ensure objectivity, this was evaluated by a separate team of experts called a blinded independent review committee.
So these folks, they had no clue which treatment each patient was getting.
Yeah, which helps avoid any bias creeping in.
Smart keeps it clean.
Absolutely. But of course, they were keeping tabs on other important stuff too, like how well those tumors were responding both in the body generally and specifically up in the brain. How long it took for the cancer to spread to the brain if it did. And, ultimately the big one, overall survival.
Yeah, it makes sense. Gotta look at the whole picture. Okay, so let's get down to brass tacks. What did they find out about how Ensartinib and Crizotinib matched up in terms of that big goal, the progression-free survival?
Here's the things that get interesting. So when they looked at all 290 patients who signed up for this study, the intent to treat population, the median progression-free survival was a whole lot longer for the folks who got Ensartinib.
Oh, nice. Can you give some numbers on that for us?
Exactly. Key efficacy outcomes showed Ensartinib led to a progression-free survival of 25.8 months compared to 12.7 months for Crizotinib.
Not bad at all, but those on Crizotinib only 12.7 months. Whoa, hold on. So we're talking about nearly double the time before the cancer starts acting up again.
Pretty much. Double the time living life without that sword hanging over your head. And to make sure this wasn't just a fluke, to give it some statistical back. They calculated the hazard ratio was 0.56 with a p-value of 0.0007, meaning it's very unlikely this difference was just random chance.
So what's that hazard ratio? Tell us in plain English.
Basically, it means that for patients on Ensartinib, the chance of their cancer worsening or even sadly of passing away during the study was almost cut in half compared to those on Crizotinib.
Wow, almost half the risk. That's got to be reassuring for patients. It's signal.
It is. It's a significant difference.
Now let's talk about modified intent to treat MITT population. Why did they do that? Did it change the results?
Good catch. The modified intent to treat population, this is a subset of patients who had their ALK-positive status double-checked and confirmed by a central laboratory who received at least one dose of the study drug. The MET population was used to assess the efficacy of Ensartinib compared to Crizotinib.
So extra verification. Why?
Well, just to be super sure to guarantee, they were looking at patients who were most likely to benefit from these drugs that specifically target that ALK rearrangement. No room for error.
Makes sense. Precision medicine.
That's the idea. And in this super refined group, the median progression-free survival for Ensartinib, they couldn't calculate it. It was not reached.
What not reached mean?
Meaning when they crunched the numbers, more than half the patients on Ensartinib were still going strong. Their cancer hadn't progressed.
That's remarkable.
It is, and for Crizotinib in the same group, it stayed the same 12.7 months.
Wow, not reached versus 12.7 months. That's a pretty clear win for Ensartinib. What about the hazard ratio in this modified group? Did it shift?
Oh yeah, it got even better. It dropped down to 0.45 and still with that super low p-value less than 0.001. So again, statistically very strong results.
Impressive. Now, they also looked at the 2-year mark, right? How many folks were still progression-free after 2 years?
They did in that MITT group. Over half of the patients on Ensartinib was a 54.2% to be precise. They were still doing great. No sign of progression at the 2-year mark. Compare that to just 36.4% on Crizotinib.
So better odds of keeping the cancer in check long term. Really good news. Now, one of the really tough things about lung cancer, especially this ALK-positive type, is its tendency to spread to the brain. Brain metastases. What did the study tell us about how these two drugs performed in that?
Yeah, good question. This is where Ensartinib, it really starts to shine. So, for patients who came into this study already having brain metastases, the confirmed intracranial response rate, meaning that tumors in the brain actually shrunk, that was 63.6% with Ensartinib. That's pretty good. Yeah, not bad, but with Crizotinib, it was only 21.1%.
Oh, wow, so way more effective at shrinking existing brain tumors.
You got it. Here's the thing, maybe even more important for you listening out there is what they saw in the patients who didn't have brain metastases at the start.
Okay, yeah, that's key. Prevention is always better than treatment.
Exactly. And with Ensartinib, the chances of developing brain metastases after a year super low, only 4.2%.
That's amazingly low.
It is, but with Crizotinib, that number jumped up to 23.9%.
The big difference?
Huge. The hazard ratio here was 0.32 with a p-value, you guessed, at less than 0.001. So again, super strong evidence that Ensartinib not only tackles those existing brain tumors better, but it's also way better at stopping them from forming in.
The first place, that's a major plus because brain metastases, they can really mess with patients' quality of life.
Exactly. It translated into a big difference in that progression-free survival for those who started the study without brain metastases.
What about the patients who did have brain metastases at the beginning? How did they fare in terms of progression-free survival?
Even in that group, Ensartinib had still showed a positive trend, although it wasn't quite as statistically significant as in the overall group or the patients without brain metastases at the start.
Trending in the right direction, right?
Yeah, their median progression-free survival was 11.8 months compared to 7.5 months for those on Crizotinib, hazard ratio of 0.55 with p-value 0.05. Not as dramatic a difference, but still suggestive of better control, even in those who already had metastases.
So, all in all, it seems Ensartinib was showing a lot of promise in tackling brain involvement, whether it's shrinking existing tumors or preventing new ones.
Absolutely, it's a game changer.
Now let's shift gears a bit and talk about how these drugs worked on the cancer and the rest of the body. The systemic response.
Okay, so the confirmed objective response rate, that tells us what percentage of patients saw their tumors shrink significance. Only within Ensartinib, across all the patients, it was 74%, and with Crizotinib, 67%.
Not a massive difference, but still better. What about in that refined MITT group?
A slight edge in the mid population, the response rates were 74% for Ensartinib and 67% for Crizotinib, still favoring Ensartinib. But here's where it gets interesting. The complete response rate, meaning the tumors completely disappeared, that was more than double with Ensartinib 14% versus 6% for Crizotinib. So, for some patients, Ensartinib was wiping out the cancer completely.
It was pretty impressive.
Absolutely.
How long did these responses typically stick around?
Well, For the MET group, they couldn't actually figure out a median duration of response for Ensartinib,
Not reached again?
exactly, which suggests those responses are lasting a good long time. They estimated that 59% of those of Ensartinib group had responses that hung on for at least 36 months.
3 years. That's remarkable. It's not just shrinking the tumors, it's keeping them away long term. Now we got to talk about the side effects with any medication. There's always that balance of benefit and risk. What did the safety profile for Ensartinib look like in this study?
Sure. So the most common side effects they saw within Ensartinib, the ones that popped up in 10% or more of the patients, included things like rash, elevated liver enzymes, both aspartate aminotransferase AST and alanine aminotransferase ALT, itching, nausea, constipation, swelling, anemia, vomiting, increased blood creatinine, and decreased appetite.
Okay, so a bit of a laundry list there, but how bad were these side effects?
Thankfully, most of them were pretty mild grade 2 or less manageable,
Good. Were there any side effects that raised particular concerns?
Grade 3 rash did show up in about 11% of the patients getting Ensartinib.
Okay, that's not ideal.
No, but even that was generally managed by adjusting the dose, and it usually cleared up within about 18 days.
Good to know. How did the overall rates of serious side effects, dose reductions, and treatment discontinuations with Ensartinib compare to those seen with Crizotinib?
Interestingly, those were pretty similar between the two groups. No big red flags for Ensartinib group, no new or unexpected safety concerns popping up.
That's reassuring. For context, what kind of side effects are typically associated with Crizotinib?
Well, the ones that come up most often with Crizotinib are liver problems, nausea, swelling, and constipation.
Okay, so they both have their potential side effects, but it sounds like the specific ones and how often they happen might be a bit different between the two.
Right, and Ensartinib seemed to have more rash issues, while Crizotinib was more linked to gut and liver stuff.
Important nuances for doctors and patients to weigh when making treatment decisions. All right, let's move on to the big one, overall survival. What did they find?
This is where things get a little tricky. The data on overall survival, it's still a bit too early to call. They haven't hit the median overall survival in either group.
Okay, so we don't know definitively if Ensartinib actually helps people live longer compared to Crizotinib.
Not yet, but they did find that the percentage of patients still alive after 2 years in that MET group was pretty close between the two treatments, about 78% for both.
So encouraging, but we need more time to see how it all plays out long term.
Exactly. And it's always worth remembering that once patients stop their initial treatment, they'll often move on to other therapies, and those can definitely influence how long they ultimately live.
Makes sense. A complex picture. Now let's put insert to them in context. How do these results measure up against what we've seen with other second-generation ALK inhibitors used as first-line treatment? Like Alectinib and Brigatinib.
Good question. So, the researchers, they actually pointed out that the median progression-free survival they saw with Ensartinib, those 25.8 months, it lines up pretty well with what's been reported from other studies comparing Alectinib and Brigatinib to Crizotinib as the initial treatment. All these newer second-generation inhibitors, they generally seem to beat Crizotinib when it comes to delaying progression.
So, and Ensartinib is right there with the other modern ALK inhibitors in terms of benefits. Anything unique about this particular study? Anything that sets it apart from those previous trials?
One thing the researchers emphasized was having that MITT group, that subset of patients whose ALK-positive status was confirmed by a central laboratory. Some earlier studies, like the ALEX trial, they required central testing from the get-go. Others allowed for local testing, which can be as a bit less reliable.
So, this central confirmation, it adds an extra layer of rigor.
It does. It helps ensure they're comparing apples to apples, making sure everyone in that group truly has the ALK rearrangement. They're creating more reliable results.
Makes sense now. The study also mentioned this concept of primary resistance. What's that about?
Yeah. So they noticed that the progression-free survival curves for Ensartinib and Crizotinib, they started diverging pretty early, like around the 3-month mark, which was sooner than what they saw in the Alectinib and Brigatinib studies.
Interesting. So, what does that really separation suggest?
Well, it might mean that Ensartinib is even more effective than those who have a kind of built-in inherent resistance to ALK inhibitors, primary resistance.
So, it's working even in those who are tougher to treat?
Possibly, but of course they are careful to point out that these different studies, they might have slightly different patient group in them, so direct comparisons are always a bit tricky. More research needed to really nail down what's driving that potential difference at a molecular level.
Definitely an area ripe for more exploration. Now, no study is perfect. What were some of the limitations the researchers highlighted?
The big ones they mentioned were the fact that we don't have the full picture on overall survival, yet still too early as we discussed. And of course, those other treatments patients get after their initial treatment stops working, that can muddy the waters when it comes to long-term survival.
Yes, right? Makes sense.
And they also pointed out that including patients whose ALK status was determined locally, not by the central lab in that overall intent to treat group that might have introduced a bit of variability, local testing isn't always as accurate. So it might have slightly skewed those efficacy results in the broader group.
Important caveats. Okay, so taking all this into account, what's the big takeaway here for our listeners?
I'd say the main message is this. Ensartinib, it's proven itself to be more effective than Crizotinib for folks newly diagnosed with this particular type of lung cancer, ALK-positive NSCLC. And it's not just controlling the cancer in the body, it's doing a stellar job in the brain too, both shrinking existing tumors and preventing new ones. And all this with like a safety profile that seems manageable and even a bit different from some of the other second-generation ALK inhibitors.
So the researchers concluded that Ensartinib is a very promising new option for first-line treatment in these patients.
Absolutely. Based on this study, it's a real contender, a compelling alternative to present to them, which has been the standard for so long, and its benefits are right up there with what we've seen from other second-generation ALK inhibitors.
Yeah, this is big news. It really shifts the landscape, and it naturally raises the question, what's next? What are the major unanswered questions? Where do we still need to dig deeper to refine our understanding and improve treatment strategies for ALK-positive NSCLC?
Absolutely, you're spot on. One of the most critical areas that needs more attention is understanding resistance mechanisms. Why do some patients not respond? And how does resistance evolve over time? Cracking that code could pave the way for more effective therapies and strategies to overcome resistance. Another key piece is treatment sequencing. With several ALK inhibitors now available, figuring out the optimal order to use them could significantly impact outcomes. Knowing when to use the right treatment can make all the difference for patients. This study serves as an excellent springboard, and it will be exciting to see how these results are integrated into clinical practice. Overall, it represents a significant advancement that could reshape the treatment approach for ALK-positive NSCLC. Notably, this research led to the FDA approval of Ensartinib on December 18, 2024, for adults with ALK-positive locally advanced or metastatic non-small cell lung cancer who have not previously received an ALK inhibitor. It's a powerful reminder of how essential continued research is in our mission to find better treatments for this disease.
This is truly groundbreaking. Dr. Pidugu, thank you for sharing your expertise and breaking down this important clinical trial for us.
Of course, Megan, keeping our listeners up to date on the latest breakthroughs in cancer research and treatment is so important.
Thanks for tuning in to OncoTrial Insights with Dr. Pidugu. If you enjoyed this episode, please subscribe and share it with your colleagues. We'll be back soon with more insights from the world of oncology research and clinical breakthroughs. Be sure to tune in next time for more oncology updates.